Fungus and bacteria can present a huge problem for all crops. They affect almost every type of plant there is. They affect yield, quality and profit. A perfect example is powdery mildew. This is a fungus that thrives in cool and humid climates, between 20 to 30 degrees Celsius. First, you'll start to notice white powdery spots on the stems and leaves of the plant. Then, if the mildew takes hold, these spots become larger and denser, spreading over the entire plant. Untreated, mildew can cause serious damage, including reduced vigor of growth development, devastation of leaves, buds and fruit, and in the worst cases, the plant will die. For growers, the economic consequences can be dire. COBE 1.2 attacks fungi through 4 different but highly effective modes of action: SAR, ISR, prevention and repulsion. SAR stands for Systemic Acquired Resistance. This kind of resistance is similar to the naturally occurring reaction when a plant is exposed to a pathogen. SAR is important to not only resist disease, but also to recover from it. COBE 1.2 first penetrates the plant through the leaves. It then triggers signaling molecules that spread throughout the entire plant. These molecules prime the genes that produce a variety of antimicrobial compounds that can attack different forms of fungi. When fungal pathogens start to attack the plant, these primed genes are switched on, acting quickly and effectively. Many proteins contribute to the defense of the plant compounds that are secreted. This combination of compounds will kill or inhibit the fungus, so it cannot grow inside the plant. ISR stands for Induced Systemic Resistance. ISR is a plant defense activation that turns on the plant's natural defenses that fight fungi. COBE 1.2 manipulates the plant to regenerate massive amounts of long chains of polypeptides and sugar molecules, as well as phytoalexins. These molecules puncture cell wands, delay maturation, disrupt metabolism, and prevent fungi production. Prevention. COBE actually promotes plant immunity and stimulates plant growth. It also selectively inhibits metabolism, preventing growth of fungal cells on the surface of the plant. Even a moderate concentration of COBE can inhibit 60 to 70% of spore growth from all fungi species. COBE has a preventative tendency to both cure and eradicate infection. The rapid translocation of anthraquinone, among them crisophenol and amodin, are known to stimulate anti-mutagenic reactions, anti-tumor, and strong antioxidant activities. Repulsion. COBE contains a naturally derived and efficient antifungal toxin. COBE focuses on the plant instead of the pest, and instead of using chemicals, it harnesses the plant's natural defense system to kill disease. You'll notice greener leaves within hours. Development. COBE contains a specially selected extract from the rhubarb plant. In nature, insects and microorganisms seem to avoid not only its foliage, but also its fruit. The roots of the rhubarb plant supply COBE with an efficient antifungal toxin. It is highly effective against spore germination of 17 tested fungi species, including 7 Alternaria species and 3 Fusarium species. In conclusion, there are many advantages to using COBE. It is a new biofungicide that employs a botanical extract with 4 unique modes of action. There is no residue and has a very low MRL index. It attacks fungi at different target sites in their metabolic pathways. It is highly effective against a wide range of pathogens, including powdery mildew, Alternaria and grey mould. Its unique methods act as a translaminar acquired resistance elicitor and has a low cross-resistance with other types of fungicides. It is compatible with other pesticides and is also harmless to Phytocellulus persimilis, honeybees, livestock and the environment in general. And finally, it's an excellent integrated pest management program compliance product. COBE 1.2 SL, a novel biofungicide.